to start going over this. So you want to fill in, we're going to go over it together and put any gaps that you missed in here. So I'm going to use this picture up here, which was from the beginning of your notes in chapter 19 to kind of go over this, because that's kind of how the chart is set up. And so your first row was pre-transcriptional regulation, so that's going on up here, what's going on before transcription occurs. So transcription, remember, is making the mRNA from the DNA. So there's some things that regulate whether or not you're eventually going to get a protein and that gene is going to be expressed at the end that occurs here. So one of the major things here has to do with chemically modifying your chromatin. So, and there's two different ways that we can do that. So, can somebody tell me one way to chemically modify your? You add acetyl groups, and as the acetyl groups added, remember chromatin is DNA wrapped around histone proteins. Are the acetyl groups added to the DNA, or are they added to the histone proteins? Histone proteins. So pre-transcriptional, the first thing that we have is, they call it histone acetylation. And so that is adding acetyl groups to your histone proteins. Now, how does this contribute to gene expression? So with histone acetylation, what happens when we add acetyl groups to the histone proteins? Nathan?
The DNA become more unwound, and it makes it easier for the DNA to be transcribed.
That's right. So here the DNA becomes kind of, I'm going to put like loosely packed around. So it's not tightly packed around the histones anymore. It's more loosely packed, and it exposes the DNA. So DNA becomes more loosely packed, exposing it, which allowing transcription to occur. So transcription increases when the acetyl groups are added to the histone proteins. The other chemical group is adding what? Roman. That has to do when we get to transcription. We haven't got there yet, so you're a little ahead of me. So we're not even to transcription yet. Another thing that can happen before even transcription can happen, Max? Yeah, the methyl groups are added to the bases of the DNA, and so this is called a DNA methylation, adding methyl groups to the bases. And in this case here, what happens during DNA methylation? And how does that contribute to gene expression? In this case here, what's going on, Gowan? And it does what? Yeah, this reduces transcription. So when those methyl groups are added to the DNA, it reduces transcription. Why does it reduce transcription? What is the methylation? That's right. So it can methylate either the maternal or paternal. I use that example with genomic imprinting. And so if one of the chromosomes is methylated, you only see only the gene from the other parent is expressed. And so what happens is when the DNA is methylated, it stays, it doesn't become loosely packed, all right, so that the DNA is exposed. So it stays kind of, so the DNA is stuck around these histone proteins, and these histones are kind of more packed together, and it doesn't expose the DNA. So the DNA is still kind of wound up more tightly. And so it reduces transcription, and the chromatin, which remember is the proteins, the histone proteins and DNA, stays packed together. Those nucleosomes stay together, and so the DNA is not exposed for the RNA polymerase to bind to it, and therefore you don't get transcription happening. Is there anything else you guys came up with before transcription? Okay, those are the two main things, all right, for this chapter. All right, so that's before. So then transcription. So now transcription, we can regulate that. Roman, how do we regulate transcription? Yeah, on the what? I couldn't. On the enhancer? Okay. The control elements. All right, and the control elements make up an enhancer. Okay, very good. So for transcription here, we can write here, as Roman said, we have activators or repressors that can bind to the enhancer region. And remember, an enhancer is made out of control elements. So you can have multiple control elements making up one enhancer. And so they can bind to the enhancer, and if it's an activator, what's it going to do? Is it going to stimulate transcription or not? Yeah, so to increase transcription, if it's a repressor, it will stop transcription. So combine to an enhancer and either stop or start transcription, depending upon which binds. Stop, that would be the repressor. Start would be the activator. And so remember that here, remember the overall, we have a gene. That gene has a promoter region. Upstream, we have control elements collectively called the enhancer. And so remember that you have different, these let's say are activators. If the activators bind, remember that every gene has a different set of control elements, and so therefore need a different combination of activators. Remember we talked about the liver cell versus the lens cell, and they have different combinations of activators that need to bind. When those activators bind, it triggers, remember, the DNA to fold on itself. Other transcription factors come and bind here, and then to the promoter region, like the TATA box, and RNA polymerase can bind and transcribe the gene. So activators turn on the gene. If a repressor, if these are repressors, if they get there first before an activator, then that's going to stop that folding, and therefore transcription is not going to occur. And so we regulate transcription in different types of cells by what type of activator and repressors are present. And every single gene has their own combination of control elements. And so this combination will turn this gene on. If these are activators, will turn this gene on. But if you add another gene, it may not turn on that gene because it's not the right combination. All right, anything else for regulating transcription? All right, good. So that's pretty much the big thing with transcription. So RNA processing. So now we make our mRNA. mRNA, remember, has introns and exons, and we cut out the introns and add a cap and a tail. So that's RNA processing. So how in the world can RNA processing contribute to the type of gene expression that happens? Lucas?
What is treated as an exon, what is an intron, so what's cut out, what isn't.
That's right. And so when you say what is treated as an intron or exon, that changes then. All right, it can change. So sometimes one part can be treated as exon, another time it can be treated as an intron. We always cut out the introns, but it's just what is treated as an exon. So RNA processing, we can change what is treated as an intron and an exon. What is that going to lead to? So we change what is treated as an as an intron or an exon. And so then what that's going to allow us to do is end up with, every time this mRNA gets processed, it can make different final mRNAs. So this leads to different mRNAs made from the same what? So you can get different mRNAs made from the same gene, right? So you have one gene, one DNA, but you can get different mRNAs. So from the same gene. And if you get different mRNAs, the mRNAs, what do? They leave the nucleus and can be used to make a protein. So you can ultimately end up with different proteins made, and therefore different gene expression. So this leads, I'm going to run here, so I'll put it down here. So different mRNAs made from the same gene, which then leads to different proteins, which means that you have different gene expression as a result of that. So you process it and you get your final mRNA. That mRNA leaves the nucleus, but before it goes to the ribosome to be translated, so you have this pre-translational modifications that can happen. So anybody, what did you guys put there? In the previous hours, a lot of people had trouble with what to put here with the Roman? Yeah, miRNA. Mi stands for microRNA. So this I talked about yesterday in the video. So microRNAs or miRNAs are small pieces, hence the name micro. They're small pieces of RNAs that can do two different things to the mRNA. So we have miRNA and mRNA. So what are the two things that miRNA can do to the RNA?
It can degrade the mRNA and it can block off its transcription.
Yeah, so it can degrade the mRNA. That's what we see here, all right, where it's going to break down the mRNA. And that's helpful in gene expression because if you have your mRNA made, then if we degrade it, it can't be made into a protein, and so therefore we're controlling the activity of that protein. So it can degrade the mRNA itself, or, as Maggie said, it can block translation. So it can make it so that the mRNA is never used at the ribosome to put together your amino acids to make your polypeptide. So it's actually blocking this part right here. And how it does that, the miRNA is a small little piece of RNA. RNA is single-stranded, and it hydrogen bonds to the mRNA. Both of these are single-stranded. So let me put up here, like you could have, let's say you have AUG here. That's part of your original mRNA. So then the microRNA would have complementary bases. So it would have UAC here. And this microRNA binds there. And now when this mRNA goes to the ribosome and the ribosome tries to translate it, it literally is a physical block, and therefore it can't translate it because it has this other little RNA stuck to it. All right, and so we must be out of time. So we'll pick this up tomorrow. All right, so we'll do the last little bit here.